Hello, my name is Leah Lambert, and today is March 5th, 2022. The first case I'm going to talk about is case 1. We have a newborn female presenting with lethargy, hypothermia, apnea. She gets blood work done, and the results of that blood work show elevated ammonia levels, urine uric acid levels are elevated, but the plasma citrulline levels are normal. We're asked to discuss the different modes that nitrogen is transferred throughout the body. So if we recall, ammonium needs to be disposed of in the liver via the urea cycle. So we need a way to get it there. So the first mode we're going to discuss is glutamine. So imagine we have a bunch of free ammonium in our cells. When this free ammonium combines with glutamate, this will yield glutamine using the enzyme glutamine synthetase. So we have to remember we need glutamine because glutamine and alanine are both non-toxic, but they're also able to pass through cell membranes, unlike that of glutamate. So now that we have glutamate, glutamine can travel to the liver and will undergo another reaction using the enzyme glutaminase. So this reaction will yield glutamate and ammonium, and now that ammonium can enter into the urea cycle. Next, glutamate undergoes a reaction using the enzyme glutamate dehydrogenase. This reaction will free the remaining amino group, and again, we see free ammonium enter into the urea cycle. So the next mode of nitrogen transfer uses alanine. Now this occurs in our muscles. So we have a bunch of ammonium in our muscles. We need to dispose of that into the liver. So we are going to need a carrier molecule. This carrier molecule that we're going to use is pyruvate. So what happens is pyruvate combines with ammonium, and in return we will form glutamate. So glutamate reacts with pyruvate to form alanine by the process of transamination. So now alanine is able to travel through the blood to the liver. So at this point it's exited our muscles going into the liver. Now it undergoes transamination again and interacts with alpha ketoglutarate. So metabolism of alanine in the liver is going to lead to the production of glutamate. Glutamate will then break down into pyruvate and ammonium, again using the enzyme glutamate dehydrogenase. So this will cause the release of ammonium into the urea cycle to be disposed of. And then what will be left is we'll have pyruvate, and that will kind of lead us into our next question, which asks us to discuss the connection between nitrogen transport and gluconeogenesis. And so nitrogen transport is connected to gluconeogenesis in the fastest state because the glucose used in the skeletal muscle was used to form the pyruvate to attach the ammonium that formed alanine. We saw that through the transfer of alanine, ammonium entered the urea cycle, and what was left was that pyruvate. So this pyruvate, through gluconeogenesis, is used to form glucose. So this glucose eventually will get shuttled back to the skeletal muscle via the bloodstream using that alanine glucose cycle. So in the next part of this case study, we're asked which enzyme is most likely to be deficient when uric acid levels are elevated, and I said the most likely one that's deficient is ornithine transcarbamylase, OTC, we'll say. This is consistent with increased production of carbamoyl phosphates, increasing pyrimidine synthesis, causing uric aciduria. So in addition to excess ammonia found in the blood, I also use the age of this patient, symptoms of lethargy, and apnea to suggest that this patient would have an OTC deficiency. So the next question had asked us what enzyme is least likely not to be impaired, and I said carbamoyl phosphate synthetase 1. And that's mainly because the data here shows high urine orotate levels, most likely caused by elevated ammonia levels found in the blood, indicating increased production of carbamoyl phosphate. So if we have an increased amount of carbamoyl phosphate, we wouldn't expect carbamoyl phosphate synthetase 1 enzyme to be impaired because that enzyme would be active. And finally, if we have a deficiency of OTC, what amino acid would be essential in this case? And I went with arginine. So arginine ultimately becomes essential because it provides an alternative pathway in the urea cycle to help remove nitrogen out of our body. So patients with an OTC deficiency cannot convert carbamoyl phosphate and ornithine into citrulline. So here we see a disruption in the urea cycle. Arginine becomes essential in the fact that it can activate the urea cycle by activating that N-acetylglutamate synthetase enzyme. And if you recall, this is the same enzyme that's required in our regulatory enzyme of the urea cycle, and that is carbamoyl phosphate synthetase 1. So here we see that arginine is essential in this pathway and a key intermediate involved in this positive feedback inhibition. And next we're going to talk about case study 3. We have a 47-year-old male. He's presenting with pain in his right toe, no history of trauma or other medical conditions are noted. Blood sample and urinalysis are done, and we have a diagnosis of gout. And this case study involves asking us what is the best explanation for the presentation of gout in this individual. And some of his results, which are going to lead us to the diagnosis, are we have high plasma urate and low urinary uric acid. So this leads me to believe that he has decreased urinary excretion of uric acid. And using the clinical manifestations of those tests, mainly the raised plasma urate levels, to explain this, uric acid circulates in the form of urate. So we have a bunch of uric acid circulating in the blood and not necessarily in the urine. It's not being able to be excreted. So in this case study, they don't mention that this man heavily drinks, but I just made a notation that this could be why this is happening, because we had slightly talked about it, how excessive alcohol consumption and the production of lactic acid can ultimately interfere with the excretion of, or compete is the word I was looking for, compete with the uric acid excretion. And I'm just going to touch on why I don't think the other ones are the diagnoses for this patient. So we know that another cause of gout can be due to the overproduction of purines, which usually manifests itself in elevated levels of urinary uric acid. And a known clinical example of this is von Gierke's disease, where we have the deficiency in the glucose 6-phosphatase. So in the disease, we have this deficiency in the glucose 6-phosphatase, and as a result of that, the pentose phosphate pathway becomes activated. This leads to an increased production of ribose 5-phosphate. And if we recall, that ribose 5-phosphate is necessary for the synthesis of nucleotides, which which in turn increases our purine basis, and that's where we can kind of see where we get that overproduction of purines. I also ruled out this because if we recall from previous units that von Gierke's disease is also a glycogen storage disease, so we would expect this patient to also present with low blood sugar, and there's no notation of that. So I had suggested maybe additional labs showing blood glucose levels in addition to a genetic history, which could be useful in determining if it was the overproduction of purines as the root cause. Next, we have the decreased salvage of purines. So the clinical manifestations that we expect to see here are high levels of urinary uric acid. A classic example of this is in Lesch-Nyhan syndrome. With the disease, we have a deficiency in the HGPRT enzyme. This enzyme ultimately plays a role in the purine recycling, and deficiency in this enzyme would increase levels of uric acid. So I ruled out this choice based on that. In addition, we had talked about how it's very common that you're going to see neurological issues if you do have Lesch-Nyhan syndrome, and this patient didn't present with any neurological issues, self-mutilating behaviors, and often a lot of these patients don't live a very long life. So the age of this man kind of supersedes what clinically we see in someone with Lesch-Nyhan syndrome. And finally, we're told that he switched recently from diet soda to regular soda, and it asks us how is this going to basically affect his gout. So normally regular soda is made up of high fructose corn syrup, and our body metabolizes that type of sugar differently than other types of sugar. So we also see that there's this direct relationship between fructose and uric acid levels. So the higher the fructose intake, we're going to expect to see higher uric acid levels. So this can occur through the way that fructose is metabolized, where hepatic ATP gets depleted, leading to promotion of uric acid and the breakdown of nucleotides. Similarly, fructose can stimulate the production of uric acid by increasing ATP degradation to AMP. So again, triggering that production of an increase in uric acid. So for this patient, we would expect that this dietary modification to have a negative impact on his gout, probably heighten his symptoms, and I would expect to see a lot more inflammation in the joints.